Yes.
Press the bell icon on the YouTube app and never miss another update.
Namaste, children. Let us continue with the topic the cell, the basic unit of life. Understood? So, in our previous class, we discussed what is cell. So, the living organisms, for the living organisms, the basic unit for the purpose of living is nothing but cell, due to the cell. So, in this class, we'll discuss the differences between plant cell and animal cell. Okay. So, you already studied in your lower classes the difference between plant cells and animal cells. So, first I will draw the plant cell. So, in plant cell, the outer membrane is cell wall, inner is plasma membrane, and this one is nucleus, mitochondria, this one is vacuole, larger vacuole, chloroplast. So these are the major organelles what we see in plant cell. Coming to the animal cell, so here we see the animal cell, nucleus, sorry, in animal cell, nucleus will be at the center, whereas in plant cell, Okay. Plasma membrane, smooth endoplasmic reticulum, rough endoplasmic reticulum, mitochondria, Golgi complex, centrioles. So like this, some organelles we see in animal cells. So the first point, as we know that cell wall is present in all prokaryotes and in some eukaryotes. In some eukaryotes andre cell wall is present only in plant cell with the prokaryotes. Prokaryotes, in all prokaryotes, the outer membrane will be the cell wall, whereas in eukaryotes, plant cell contain only the cell wall, it is absent in animal cell. Okay, here cell wall is present. So here cell wall is absent. Here what will write? Cell wall is absent. Understood? Other difference between these two is, see here we see large central vacuole. So large vacuole is present. Okay. Here we see very small vacuoles are present, or they may be absent also. Small vacuoles are present. Sometimes vacuoles are absent in animal cells. The other thing is, due to the presence of large vacuole, nucleus is pushed towards the periphery. Understood? So why the nucleus is present towards the periphery? Nucleus is towards periphery. It's towards the cell membrane. Understood? Whereas in animal cell, nucleus is present at the center. Nucleus is at the center of cell. Understood? So one more difference is, see here I wrote centrioles here. Understood? Centrioles are absent in plant cell. Centrioles are absent in plant cells, whether centrioles are present in animal cells. They helps in cell division, during cell division. And one more, so chloroplast, which is responsible for autotrophic nutrition in plants, is present in plant cell. Chloroplasts are present. Understood? They are present in plant cell, but here the chloroplasts are absent. So fifth point I'll write here, chloroplasts are absent. Understood? So these are the differences between plant cells and animal cells. So in plant cells, we see the presence of cell wall, but the cell wall is absent in animal cell. Due to the presence of cell wall, plant cells have definite shape, whereas animal cells does not have a definite shape. And one more difference is presence of large vacuole in plant cell. The smaller vacuoles are present in animal cell. If they are present, they are smaller, otherwise they are absent. Then nucleus is pushed towards the periphery due to the presence of central large vacuole. Here, as the central large vacuole is absent, nucleus is present at the center of a cell. Other one is centrioles are absent in centrioles are absent in plant cells, but they are present in animal cells, which helps during cell division, which are very important, which play a very important role in the cell division. So these are the differences between plant cell and animal cells. Now we'll discuss the functions of each organelle present in both plant cell and animal cells. Understood? So coming to the cell wall. So we already know that cell wall is present only in plant cell. They are present only in plant cell. Present only in plant cell. So like this, the structure. So we see middle lamella between the cells. So what I am writing this color, this represents a middle lamella present between the primary cell wall and secondary cell wall. So in detail we study in your regular classes. In this class just we'll go with the few things in the function of cellular organelles. Okay, so cell wall. So we know that cell wall is present only in plant cell, and also we see the cell wall in prokaryotes. It is present, its outer layer in prokaryotes. And composition of cell wall in prokaryotes is different. Understood? Composition of cell wall in prokaryotes is different. So in the plant cell also the composition differs from one cell with the other cell. Majority in plant cell, the cell wall is made up of cellulose and hemicellulose, pectin, etc. So the composition of cellulose, the composition of cell wall is cellulose, hemicellulose, pectin, etc. We see this in many plant cells. The composition of cell wall will be cellulose, hemicellulose and pectin. Coming to the algae, there we see galactans, mannans as a composition of cell wall. Understood? So here other point we'll write, the cell wall is non-selective. Non-selective means it allows all the substances to move in and out. Whereas coming to the cell membrane, inside the cell wall, what we see cell membrane is selectively, or plasma membrane is selectively permeable, but it is non-selective. And other function is it gives definite shape to a cell. Understood? The middle lamella present between the cell wall connects the two cells, connects the two plant cells. So the middle lamella middle lamella connects two adjacent cells. Which is made up of calcium pectate. It is made up of calcium pectate. It is made up of calcium pectate. If the two cells will separate each other antandre, there we see the dissolution of middle lamella. Two plant cells are connected with each other with the help of middle lamella. ಎರಡು ಸೆಲ್ ಸಪರೇಟ್ ಆಗ್ತಾ ಇದೆ ಅಂತ ಅಂದ್ರೆ there we see the dissolution of middle lamella. There is absence of middle lamella. So that middle lamella is made up of a substance called calcium pectate. And we see plasmodesmata between the cell wall and middle lamella. We see a junctions called plasmodesmata which helps in connection between the two cells and allow the passage of movements between one cell with another cell. It allows the movement of substances between one cell with another cell like this. So it allows the movement between two adjacent cells. So this is about cell wall. Cell wall is present in all prokaryotes, and in eukaryotes it is present only in plant cell. Cell wall is made up of cellulose, hemicellulose, pectin, etc. The composition of cell wall also varies from one plant cell with another plant cell. It is non-selective. That means it allows all the substances to move in and out of the cell. Due to the presence of cell wall, plant cell have a definite cell shape, and two adjacent plant cells are held together with the help of middle lamella, which is made up of calcium pectate. If the cells are going to be separate, that is mainly due to the dissolution of middle lamella. ಇವಾಗ ಮರದಿಂದ ಹಣ್ಣು ಕಾಯಾಗಿ ಹಣ್ಣಾಗಿ ಉದುರುತ್ತೆ ಅಂತ ಅಂದಾಗ ಆ ಸೆಲ್ಸ್ ಇಂಟ್ಯಾಕ್ಟ್ ಇರುತ್ತಲ್ಲ ಮರದ ಜೊತೆಗೆ, ಅವಾಗ ಆ ಸೆಲ್ಗಳ ಮಧ್ಯೆ ಏನಿರಲ್ಲ, ಮಿಡಲ್ ಲ್ಯಾಮೆಲ್ಲ ಡಿಸೊಲ್ಯೂಷನ್ ಆಗಿ ಆ ಹಣ್ಣು ಮರದಿಂದ ಕೆಳಗೆ ಬೀಳುತ್ತೆ. ಸೊ that is mainly due to the middle lamella. The adjacent cells are held together with each other in plant cells due to the presence of middle lamella. And we see the connections between two plant cells with the junctions called plasmodesmata. So this is about the cell wall. Next we'll go with the plasma membrane or cell membrane. So plasma membrane, it is the outermost layer in animal cell, whereas in plant cell we see inner to the cell wall. So inner to the cell wall we see this plasma membrane. Understood? So what's the function of plasma membrane? Plasma membrane is selectively permeable. It is selectively permeable. That means, as I told, cell wall is non-selective. It allows all the substances to move in and out. ಆದರೆ ಇಲ್ಲಿ plasma membrane is not allowing the substances, every substances to move in and out. It selects some substances. ಯಾವುದನ್ನು ಸೆಲೆಕ್ಟ್ ಮಾಡ್ಕೊಂಡಿರುತ್ತೆ ಅದನ್ನಷ್ಟೇ ಒಳಗಡೆ ಮತ್ತೆ ಸೆಲ್ ಒಳಗಡೆ ಸೆಲ್ ಹೊರಗಡೆ ಹೋಗೋಕ್ಕೆ ಅಲೋ ಮಾಡುತ್ತೆ. ದಟ್ಸ್ ವೈ plasma membrane is also called as selectively permeable membrane. It is made up of lipids, bilayer of lipids. Cell membrane is made up of lipids, and it allows the movements either by diffusion or by passive transport or by active transport. Movement of substances between one cell with another cell will occur either by diffusion or passive transport or active transport. Diffusion means they does not require any energy, they just move from one cell to another cell. Passive transport occurs along with the concentration gradient. That means the substances will move from higher to lower concentration. That come under passive transport. Even the diffusion also come under passive transport. There also we see the movement of substances without using any energy. Whereas active transport, it utilizes ATP. That means here the substances will move from its lower concentration to higher concentration. Lower to higher concentration. That's why they require energy. ನಾವಿವಾಗ ಮೆಟ್ಲು ಹತ್ಕೊಂಡು 5th ಫ್ಲೋರ್ ಹೋಗ್ಬೇಕಂದ್ರೆ ಲೋವರ್ ಟು ಹೈಯರ್ ಹೋಗುವಾಗ ವಿ ನೀಡ್ ಎನರ್ಜಿ. ಹಾಗೆ ಮೇಲಿಂದ ಹೇಳ್ಕೊಂಡು ಬರುವಾಗ ತುಂಬಾ ಈಸಿಯಾಗಿ ಹೇಳ್ಕೊಂಡು ಬರ್ತೀವಿ. ಸೊ ದ ಮೂವ್ಮೆಂಟ್ ಆಫ್ ಸಬ್ಸ್ಟೆನ್ಸಸ್ ಫ್ರಮ್ ಹೈಯರ್ ಟು ಲೋವರ್ ಡಸ್ ನಾಟ್ ರಿಕ್ವೈರ್ಡ್ ಎನಿ ಎನರ್ಜಿ. ಅದಕ್ಕೆ ನಾವು ಪ್ಯಾಸಿವ್ ಟ್ರಾನ್ಸ್ಪೋರ್ಟ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಆಕ್ಟಿವ್ ಮೀನ್ಸ್ ಇಟ್ ಯುಟಿಲೈಸಸ್ ಎನರ್ಜಿ when the substances move from lower to higher concentration. Understood? So the plasma membrane is selectively permeable membrane. It is seen in both plant cell and animal cell, made up of lipids. It allows the movement of substances by a process of diffusion or passive transport or active transport, and even the movement of water occurs by a process called osmosis. What is osmosis? Movement of water from its higher concentration to lower concentration through a selectively permeable membrane. So that also is one of the function under plasma membrane. Understood? This is about plasma membrane. Next, we'll go with a structure called chloroplast. So the chloroplast is a double membrane organelle. It is having two membrane, outer membrane and inner membrane. Outer and inner membrane. So here we see the arrangement of chlorophyll pigments in a these are thylakoids arranged in a pattern called grana. Piles of thylakoids are called grana. So this is stroma lamellae which connects to grana, and we see this cytoplasm called stroma. So what's the function of chloroplast? Chloroplast, it is present only in plant cells, contain a pigments called chlorophyll. Pigments called chlorophyll, which helps in photosynthesis, trapping sunlight. It helps in trapping the sunlight and converting light energy into chemical energy. Photosynthesis. So here, by using carbon dioxide and water in the presence of sunlight, this process occurs in chlorophyll to form a organic compound called C6H12O6, that is glucose plus O2. Understood? Which is an exothermic reaction. So like this, in chloroplast we see the formation of glucose. So that's due to the presence of chloroplast in due to the presence of chloroplast in plant cell, they are autotrophic in nature. That means they'll prepare their own food. They'll not depend on other living things for their food. As we lack this chloroplast, animals always depend on other living things for their food. So this is about chloroplast. Next we'll go with mitochondria, which is seen in both plant cell and animal cell. This is also a double membrane structure having outer membrane and inner membrane. Here the inner membrane shows enfoldings called cristae. It shows enfoldings called cristae, where we see the oxidation of glucose. It is having a F1, F0 particles where we see the oxidation of glucose. That means breaking of glucose. C6H12O6 in presence of oxygen, it gives rise to CO2 plus H2. See, just opposite to photosynthesis. Understood? Just opposite to photosynthesis with the liberation of ATP. So that's why mitochondria is called powerhouse of the cells, because where we see the liberation of ATP, adenosine triphosphate, which we use to do our daily activities. Understood? So this is about mitochondria. One thing we have to remember about both these structures, mitochondria and chloroplast, because they have their own DNA. So this is a cell. This is a cell. So inside the nucleus we see a chromatid DNA. Apart from this, the mitochondria, it is also having its own DNA, and chloroplast is also having own DNA. They have their own DNA apart from the genomic DNA, and these two structures have 70S ribosomes. In prokaryotes, we see the ribosomes. They are 70S. Same ribosomes are seen in mitochondria and chloroplast. Inside the mitochondria and chloroplast. Whereas in eukaryotes, chromosome is of 80S. Eukaryote cell chromosome ಎಷ್ಟು size ಇದೆ? 80S ಇದೆ. Outside the cell, we see in cytoplasm of the eukaryote, we see 80S. But inside the mitochondria or inside the chloroplast, we see ribosomes with 70S. Understood? Ribosomes with 70S is present inside the mitochondria and chloroplast, whereas in cytoplasm of the cell, eukaryotic cells, it is 80S. So this is about mitochondria. As they produce ATP by the oxidation of glucose in the presence of oxygen, they are called as powerhouse of the cells. They are called as powerhouse of the cells. This is about mitochondria. Next, we'll go with endomembrane system. Endomembrane system include endoplasmic reticulum, lysosomes, vacuoles, and Golgi complex. These all come under endomembrane system. Why all these four organelles come under endomembrane system means the functions are coordinated. All the 4 functions are coordinated with each other. That's why they all come under endomembrane system. So coming to the endoplasmic reticulum, we know that attached to the nuclear membrane, we see the endoplasmic reticulum. There are 2 types of endoplasmic reticulum. Rough endoplasmic reticulum, when we see the presence of ribosomes on its surface, like this. If ribosomes are present, it is called rough endoplasmic reticulum. If ribosomes are absent, it is called as smooth endoplasmic reticulum. Rough endoplasmic reticulum helps in protein synthesis, whereas smooth endoplasmic reticulum helps in lipid synthesis. This is the difference between rough endoplasmic reticulum and smooth endoplasmic reticulum. Understood? So, the endoplasmic reticulum is present in both plant cell and animal cells. If they have ribosomes on its surface, they are called as rough endoplasmic reticulum, which mainly involved in protein synthesis. If the ribosomes are absent, they are called as smooth endoplasmic reticulum, where we see the synthesis of lipids. So, this is about endoplasmic reticulum. Next, we will go with lysosomes. So, coming to the lysosomes, they contain enzymes, hydrolases. They contain enzymes called hydrolases. ನೋಡಿ, enzymes always end with ASC ಅಂತ ಹೇಳಿ. They contain enzymes called hydrolases like proteases, lipases, amylases. ಹೀಗೆ enzymes are present in lysosomes which are active at acidic pH. They are active at acidic pH. Proteases helps in digestion of proteins. Lipases helps in digestion of lipids. And amylases helps in digestion of carbohydrates. So all the enzymes are present in lysosomes. Understood? So lysosomes contain hydrolases. That means hydro means water. To break protein, lipids and carbohydrates, it required water. ಅದಕ್ಕೆ ಅಲ್ಲಿ hydrolases. Enzymes required water to digest protein, lipids and amylases. Understood? So all the enzymes together we call it as hydrolases, which are active at acidic pH. So the next, coming to the vacuole. So vacuole, we already know that in plant cell, vacuole occupies maximum space. Larger vacuole is present in plant cell. The membrane of the vacuole in plant cell is called tonoplast. Membrane of the vacuole in plant cell is called tonoplast. Whereas in animal cell, vacuoles are absent, or if they are present, they are very smaller in size. In plant cell, vacuoles, they store excretory products or unwanted substances which are not useful to cells are stored inside the vacuole. That's stored inside the vacuole. So that is about vacuoles. Then coming to Golgi complex. Golgi complex mainly helps in synthesis of glycoproteins and glycolipids. Glycoproteins and glycolipids. Understood? So this is about endomembrane system. Under endomembrane system, we see the four organelles: endoplasmic reticulum, lysosomes, vacuoles, and Golgi complex. Why they all come under one system? Because the functions of each other are coordinated. And among these, endoplasmic reticulum is seen in both plant cell and animal cells, which again divided into two types: smooth endoplasmic reticulum and rough endoplasmic reticulum. Rough endoplasmic reticulum is having ribosomes on its surface, hagagi it helps in protein synthesis. Smooth endoplasmic reticulum does not have ribosomes on its surface and it mainly involved in lipid synthesis. Lysosomes contain enzymes to digest protein, lipid and carbohydrates. Vacuoles are larger in size in plant cells which store unwanted or excretory products which are not useful to the cell, and they are very small or absent in animal cells. Golgi complex are present in both the cells which mainly involved in synthesis of glycoproteins and glycolipids. This is about endomembrane system. Coming to the ribosomes, this is non-endomembrane system. Athwa, we'll go with it's non-membrane bound organelle. Non-membrane bound organelle, which is mainly involved in protein synthesis. Ribosomes are exist in two forms. One is larger unit, another one is smaller unit, held together with the help of magnesium. And in prokaryotes, we see 70S ribosomes, whereas in eukaryotes, we see 80S ribosomes. S represents Svedberg's unit. S under Svedberg's unit anta, which tells us sedimentation coefficient. Sedimentation coefficient, nothing but it tells us density and size of that organelle. Density and size of organelle. This is about ribosomes. It is non-membrane bound organelle present in all prokaryotes and eukaryotes, but in prokaryote it is 70S Svedberg's unit. Aden represent ಮಾಡ್ತಿದೆ. It is giving us sedimentation coefficient. Hangandre eno density ಅಂದ್ರೆ ತೂಕ. It gives us density and size of the organelle. In eukaryotes we see 80S. In both prokaryotes and eukaryotes, the main function of ribosomes is nothing but protein synthesis. That's why ribosomes are called as protein factories of cells. Why they are called as protein factories? Because they involve in synthesis of proteins. So protein factories of cells. This is about ribosomes. Next we'll go with nucleus. We know that in plant cell it is pushed towards the periphery due to the presence of central large vacuole. So nucleus, Robert Brown discovered the nucleus. Nucleus is surrounded by a nuclear membrane. Inside the nuclear membrane, we see a nucleoplasm. which contain nucleolus and chromatin network. Understood? This chromatin, the cell which is not involved in cell division, ಯಾವ cell division involve ಆಗಿಲ್ಲ, ಅಲ್ಲಿ it is present in a thread like fashion. It is called chromatin. This chromatin condenses like this. ತನ್ನನ್ನು ತಾನೇ ಸುತ್ಕೊಂಡು ಏನು ಮಾಡುತ್ತೆ, it condenses like this and form a structure called chromosomes, and it forms a structure called chromosomes during cell division. Form a structure called chromosomes during cell division. Like this. So this is centromere. Based on centromere, chromosomes are again different types: metacentric, submetacentric, telocentric, acrocentric, like that. So that we'll study in our regular classes. So in this class, nucleus is present in both plant cell and animal cell. In eukaryotes, nucleus is having a nuclear membrane. Inside that we see a cytoplasm called nucleoplasm, which contain nucleolus. Nucleolus mainly helps in protein synthesis, and other structure present inside the nucleoplasm is chromatin, which is always present in thread like manner. During cell division, it condenses, tanitane coil agi it forms a structure called chromosome. So this chromatin is nothing but which contain DNA. It is responsible for carrying the parental characters to their offsprings. So this is very important for heredity. DNA is responsible for carrying parental characters to their offsprings. Understood? So this is about nucleus. And as I told, centrioles are present only in animal cell, that helps in cell division. So this is about some organelles functions which are present in eukaryotic cells. Understood? Once we revised what we discussed in this class. So we discussed the difference between plant cell and animal cell. We know the difference between plant cell and animal cell as the cell wall is seen only in plant cell, chloroplasts are present only in plant cell, and large vacuoles are present only in plant cell. Coming to the animal cell, centrioles are present in animal cell and smaller vacuoles are present in animal cells, and lysosomes also present in animal cells. Then cell wall is composed of cellulose, hemicellulose, pectins. Two plant cells are held together with the help of a structure called middle lamella, which is made up of calcium pectate. Cell wall is non-selective. It allows all the substances to move in and out. And coming to the plasma membrane or cell membrane, it is seen in both plant cell and animal cell. It is made up of bilayer of lipids, which is selectively permeable. That means it allows some selected molecules to move in and out. And it allows either by diffusion process or active transport or passive transport. Then we discussed mitochondria, which is called as powerhouse of the cells, because inside the mitochondria, the oxidation of glucose takes place, which releases ATP. And mitochondria, that's why mitochondria is called powerhouse of the cells, and it has its own DNA and 70S ribosomes. Chloroplasts are present only in plant cells. Due to the presence of chlorophyll pigment, it is able to synthesize glucose in the presence of sunlight by using carbon dioxide and water. Due to this, it mainly helps in autotropism in plants. Even chloroplast is having its own DNA and 70S ribosomes. Coming to endomembrane system, it includes 4 organelles. Yavadu endoplasmic reticulum, Golgi complex, vacuoles, and lysosomes. Endoplasmic reticuli again are of 2 types based on presence or absence of ribosomes. If ribosomes are present, they are called as rough endoplasmic reticulum. They mainly helps in protein synthesis. Ribosomes illa andaga, it is called as smooth endoplasmic reticulum, which helps in lipid synthesis. Then coming to lysosomes. Lysosomes contain set of enzymes called hydrolases, which helps in digestion of protein, lipid and carbohydrates. They are active at acidic pH. Vacuoles contain excretory products or unwanted things for the cell are stored in the vacuole. The membrane of the vacuole is called tonoplast. Golgi complex mainly helps in synthesis of lipids like glycolipids and glycoproteins. The next we discuss nucleus. Nucleus again in prokaryotes it is DNA is not surrounded by nuclear membrane. That's why we call it as naked DNA. Whereas in eukaryotes the DNA is surrounded by a membrane called nuclear membrane. And inside the nuclear membrane we see nucleolus and chromatin network inside the nucleoplasm. So nucleolus mainly helps in protein synthesis, whereas chromatin network forms a chromosomes during cell division by either coiling it on itself or by condensation process. Understood? This is about the functions of organelles which are present in plant cell and animal cells. So in next class we'll go with the chapter the living world. There we'll see the difference between what's the major difference between the living and non-living things and some of the defining characters which are present only in living things that we'll discuss in our next class. Okay. Thank you.